Fran, thank you very much for the introduction and the invitation and organising this symposium today. So Henrik posed a question, is all of the answer going to be in genetics? And I'm going to say definitively at the beginning of this talk, no, the answer is not all going to be genetics. I'm sure genetics will have a role, but that's going to be context dependent. Now, it's an interesting time, and it's an interesting time because there's been huge technology developments in genetics which are still going on. And the volume of genetic sequencing data is really, certainly in a clinical context, is increasing at an exponential rate. 5 years ago, it was seen as a great achievement to generate a whole genome sequence for a handful of patients, whilst now the NHS in the UK is embarking on doing whole genome sequencing on tens of thousands of patients. So we're actually awash with sequencing data. Some of the difficulty is analysing this data just at a bioinformatic level, and I would say probably even more of a challenge is how do you relate that sequencing data to phenotype. Now, all of you in the audience here today, there will be variation in your genetic code. There will be some variation, for instance, single nucleotide polymorphisms, which are actually quite common within the general population. There will be some variation which is much rarer, and so, for instance, some gene variants are virtually never present in the general population. Although, again, the advances in genetic technology are telling us actually if you sequence a person's whole genome, many of them will have a significant number of mutations that you would predict would have a major impact on protein function, except they don't lead to any significant phenotypic problem. And so this comes back to this issue of how do we relate genotype to phenotype, and here we're talking about pain today. Of course, pain is a particularly challenging phenotype, and that's because it has many different aspects, an affective component, a discriminative component. Many people use different pain descriptors, whether it's a burning pain or electric shock-like pain, or whether you're looking at the response to particular sensory modalities. So I think we have some real challenges here. And of course, I'm going to tell you at the beginning of this talk about some rare gene variants that have a big impact on function and lead to Mendelian pain disorders, but the much more common clinical problem, which Henrik so nicely described, would be that someone's fine and then they're exposed to some kind of environmental exposure. Henrik performs a hernia repair, and it's actually that environmental stressor that leads to a chronic pain state. And of course, that's an interaction between genes and environment and selective vulnerability. Of course, when we're doing these studies, and I will come back to this nearing the end of this talk, we may use intermediate phenotypes or endophenotypes. It may be, for instance, in this patient that has erythromelalgia, a physical manifestation of the underlying biology, which would be a flare response in her skin. It may be a particular pattern of sensory profile on quantitative sensory testing, or it may, for instance, be an imaging biomarker. And the way I'm going to structure this is I'm going to start talking with what I think are rare conditions, and in a way they're an easier problem for me to pose to you, which is high impact variants that cause Mendelian pain disorders. And I think there we have excellent evidence that the gene variants are causing that disorder, and they give us great insight into the biology of the nervous system. I'm then going to touch on the more difficult problem, which is acquired pain disorders, which are very much more a polygenic issue of interactions between multiple genes and the environment, leading to a chronic pain disorder, and how we're just beginning to try and tackle that problem. So I think in terms of monogenic pain disorders, there's clearly an interest in studying those patients that don't feel any pain. So I'll just give you an example of a patient of mine in Oxford, and if you want to know why people need a system to detect tissue injury, then you just have to look after these patients. So this is a 26-year-old man, and he's never experienced pain since birth, and his growth was stunted because he developed so many fractures during childhood. He destroyed every single growth plate in the long bones in his body. These patients will tell you stories such as this gentleman heard that there was a financial reward if he lost a tooth and it was left under his pillow, and he promptly went to the shed, took his father's pliers out and pulled out a number of his own teeth. And this example here shows you, if you look at the genetic pedigree, that actually he's the one with the arrow pointed to him. His parents were unaffected. He has a younger sister that's affected, and another sister that died at a young age because of unrecognized sepsis, because she could not detect tissue injury. And if you examine this patient, I've told you that he's short, he couldn't smell, and I'll explain there is a biological reason for that later. And if you were to test large fiber functions, such as stroking the skin, proprioception, vibration, it's all completely normal. But whether you apply a noxious stimuli to any part of the body, whether it's chemical, thermal, or mechanical, he never experiences it as painful. And what's interesting about these patients is if you were to go on and do a skin biopsy, which we did in this patient, and you were to stain it for nerve fibers, which are shown in red, you will find that nerve fibers are there. So it's not a problem that the nociceptive neurons have not developed. They have developed, they're just dysfunctional. And it was Jeff Woods and James Cox working with Jeff Woods in Cambridge that provided us with the genetic basis of this condition. And they studied a consanguineous pedigrees within Pakistan, and they found that this condition is due to loss of function mutations in an autosomal recessive fashion in a gene called SCN9A that encodes a voltage-gated sodium channel called NAV1.7. And NAV1.7 is expressed within the peripheral nervous system, expressed particularly within sensory neurons and autonomic neurons, but it's also expressed in olfactory neurons, and that leads to this combination of insensitivity to pain as well as anosmia. And this patient I was just telling you about, he's a compound heterozygote, so on one allele there's a premature stop codon, which means that a truncated protein is formed, and on the other allele there's a frameshift mutation, also leading to lack of protein expression. And the essential hypothesis is that you're not producing functional NAV1.7, and NAV1.7 is essential for the detection of tissue injury stimulus and the perception of pain. Actually, it's not a trivial question to answer whether we think that NAV1.7 is essential at the very peripheral terminals during early transduction events, whether it's required for the long-term transmission, long-range transmission of action potentials along the axon, or actually whether it may have a role in neurotransmitter release, and there's some evidence for all of those factors. Increasingly in these monogenic pain disorders, we're trying to use human cells as a model system to study some of these events, and just to give you an example of that, so now we can take a skin biopsy, reprogram it, so that we generate induced pluripotent stem cells, which are cells that can, with the right conditions, differentiate into any cell type in the body, and then using a particular differentiation protocol, generate neurons that look very much like sensory neurons. So these are now neurons in a dish, and the green is showing axons, the red is their cell bodies, and they're expressing a marker which is selectively expressed within sensory neurons. And the good news is that if we patch clamp these neurons, they can generate action potentials, and they've got a nice shoulder and a broad action potential, which are the characteristics that we would expect of nociceptors. So this is now a model system where we can start exploring how a genetic defect is causing a biological problem. And in fact, when we do this and we inject current into these neurons and we compare neurons that we've developed from that patient with congenital insensitivity to pain to neurons that we've generated from control subjects that have normal pain sensibility, there's a clear hypoexcitability in the patient, and he's finding much fewer action potentials in response to current injection. I have to say that excitability is not completely ablated, so there is still some response, it's just markedly reduced, and I still think it's an open question as to whether NAV1.7 may not just be having roles in terms of action potential propagation, but for instance in neurotransmitter release. So I'm just going to show you the flip side of that coin with another rare condition, but gives us insight into biology, and is another Mendelian pain disorder. And that's the condition called inherited erythromelalgia. And erythromelalgia really is just a symptom complex of painful red extremities. And usually this pain is exacerbated by warmth. So this patient that I'm showing you here, she's standing in her shower and she's putting her feet in cool water. And that's because often cooling can help relieve the very disabling pain that these patients experience. This particular patient, she has been in pain actually ever since birth. She was born with red feet, and she's always had painful red feet, and she gets very severe pain in the context of a warm environment. And it's an autosomal dominant pedigree, so it's a single allele causing this problem. And in fact, the patient that I've just shown you, she has a mutation in the same ion channel. She has a mutation in NAV1.7, but now this is not a loss of function, this is a gain of function mutation. And she's got a mutation in this linker domain between S4 and 5 in domain 2, and this is a hotspot for mutations in NAV1.7. And we should give great credit to Steve Waxman, who has really driven the understanding of the biophysics as to how these mutations are associated with this clinical syndrome. And in fact, we now know that there's a large number of mutations within NAV1.7 that can cause inherited erythromelalgia. And the general theme is that these mutations, with a few exceptions, are predisposing this ion channel, making it more likely to activate. So there's a hyperpolarizing shift in the voltage dependence of activation. And this is just a picture I've taken from one of Steve Waxman's papers, where in the top panel, he's injecting current into neurons expressing a wild-type NAV1.7, and in the bottom panel, he's injecting current into neurons expressing NAV1.7, which is mutated, and the mutation of a patient with erythromelalgia, and you can see there's much greater action potential firing in the context of the mutated ion channel. So we have a nice biophysical correlate of the clinical syndrome, which relates to that mutation. And actually, it's now becoming clear that in terms of monogenic pain disorders, many of these are impinging on the way that nociceptors work, both in the transduction and transmission machinery. So I haven't had time to go into it in detail today, but I had a very pleasant trip to Medellin in Colombia, where we found a family that had gain-of-function mutations in an ion channel called TRPA1, which is expressed on, which is important for transduction, particularly of noxious cold stimuli and in environmental irritants such as components of tear gas and mustard oil. And we found a nice correlate in those patients which had an episodic pain disorder, but not only did they have hypersensitivity of the channel when it's heterologically expressed, but actually when we gave the agonist to those patients in a kind of experimental medicine paradigm, they also showed hypersensitivity to agonists at this ion channel. We've just been discussing NAV1.7, which is an excellent example because if you lose function in this ion channel, you're insensitive to pain. If you have mutations that cause gain of function in this ion channel, it can cause a number of Mendelian pain disorders. I've given you one example, which is erythromelalgia, but there are other examples such as another set of mutations can actually cause degeneration of small fibers and a small fiber neuropathy. And there are other voltage-gated sodium channels which are expressed within nociceptors, such as NAV1.8 and NAV1.9. And again, we're beginning to find that there's a preponderance of gain-of-function mutations in these ion channels associated with small fiber neuropathy. Finally, we have evidence we haven't published yet that some patients, there's a human condition associated with mutations in Kv1.1 and potassium channels that are breaks in excitability, and these mutations lead to impaired potassium channel function, and actually some of those patients which have episodic ataxia also develop neuropathic pain. So actually we're seeing a real cluster of these Mendelian pain disorders impinging on targets which are expressed within nociceptors that are ion channels important for the regulation of excitability. And you could say, well, how about now moving to kind of common clinical problems, such as painful diabetic neuropathy. So we've just started this work, but we have quite a large cohort of patients, almost 400 now, with both painful and painless diabetic neuropathy. And when we start looking for mutations in NAV1.7, we do in a small percentage of patients. So this is not going to be a common predisposing cause to developing pain in diabetic neuropathy. We're talking in 5% of patients or less. We're beginning to see some of those mutations that have been previously described, either in erythromelalgia or in small fiber neuropathy, being present in those patients. So this would be an example of rare variants which have a high impact being present in a small proportion of patients, contributing to quite a common clinical problem, which is acquired painful diabetic neuropathy. You may ask, is there any way of identifying those patients? And I haven't found one so far. So all of these patients have had profiling using quantitative sensory testing, and generally the finding that which you may expect in diabetic neuropathy is hypofunction and reduced sensibility to applied sensory stimuli across multiple different modalities. And actually, we pretty much find exactly the same thing in those patients that do have the mutations in NAV 1.7. So this would be something of a challenge for personalized medicine if you were going to try and pick those patients on their clinical phenotype as to who you would be doing the sequencing on. So I hope I've convinced you, and I think the evidence is very good, that you can get some high-impact rare mutations that have effects on ion channels and cause Mendelian pain disorders. What about this much more common problem, and really the problem that Henrik posed to you, which is those more common acquired pain states, such as post-surgical pain? Well, as I implied in my introduction, and if you look at this graph, which is taken from a review in Nature, which looks at the relation of effect size of a gene variant related to how common that variant is within the population. So there are rare alleles, rare gene variants, which are virtually either present in a very low proportion of people or virtually absent from the normal population, which have a very big impact. So these would be the disorders I've just been telling you about would be within that spectrum. Common diseases such as hypertension, or you could argue migraine, or you could argue post-surgical pain, are likely to be due to multiple genes with a small effect interacting with the environment. And that would be down on that bottom right corner. And these are much more challenged to try and ascribe pathogenicity. And traditionally, a genome-wide, you could either use a candidate gene approach, or you could use genome-wide association studies to try and get a handle on this genetic problem. And those kind of studies, certainly candidate gene studies, have been a number within the pain fields, which have provided very interesting outcomes. We've been interested in the idea of performing genome-wide studies, really using an intermediate step. So I think taking patients is extremely challenging, partly because of the variation and we've heard much of that variation, for instance, in surgical practice that you have to deal with. One way of dealing with this is to use experimental pain models as a kind of intermediate step. And Henrik's given you some evidence already that, for instance, in certain contexts, the heat pain threshold can be predictive of those patients that would go on to get persistent pain post-surgery. So could we employ experimental pain models? Well, is there evidence of heritability within those experimental pain models? And a good friend and colleague of mine, Steve McMahon, working with Tim Spector in the Twins unit in the UK, and Francis Williams was talking with data from that unit earlier at this meeting, has looked at the heritability employing experimental pain model in the Twins. And they were using a protocol where they had a thermal burn protocol and looking at the sensitization following that thermal burn protocol. And by comparing looking at comparing concordance between monozygotic and dizygotic twins, they can get an estimate of heritability. And actually, some of these experimental pain measures, such as punctate hyperalgesia, really had a significant heritability of around about 55%. So that is evidence that genes are playing a part in the variation within the population in response to that experimental pain model. So in fact, I'd already worked with Andres Ruiz Linares, who has a collaboration between UCL and Columbia, and we instituted an experimental pain model in collaboration with Andres. And the reason that we did that, well, there were a number of reasons. Well, we did this in Medellin, and this is a picture of the University of Antioquia in Medellin. And Andres has been interested for quite some time, and this is a picture from one of his papers, in genetic intermixing and the origin of populations within South America. And that's because it's an interesting issue whereby it's a mixed genetic population with African lineage, the native Indian lineage and European lineage, and they've been looking at multiple sites at the impact of various gene variants on phenotypic features within these populations. And in fact, they already have a large amount of sequencing data using for genome-wide analysis of these populations, and what we needed to do is bring these patients back and re-phenotype them using an experimental pain model. And just to give you an idea of this approach, So we could look at their baseline thresholds and have a training session on one arm. So we'd look at, for instance, heat pain, cool detection, warm detection, pressure pain thresholds. We could then institute this on the arm that we were going to do our test on, and then we gave a sensitizing stimulus because we wanted to see what happens to the sensitized nervous system. And because we wanted to develop an efficient protocol as possible, we gave mustard oil, which I've already told you is an agonist of TRPA1, and it leads to sensitization of the nervous system, probably with both a peripheral, and Henrik was talking earlier, also a central component. And this is my arm here, and in the central circle is where we've just given mustard oil, and around that you'll have a flare response as you get antidromic activation of nociceptive afferents. And then you can map out the sensory dysfunction. So there will be a region of punctate hyperalgesia, increased sensitivity to a punctate mechanical stimulus, which essentially relates to central sensitization. But also we can go back and measure. We already did the mechanical pain thresholds and heat pain thresholds at baseline. We can look at the sensitization following this. So this is an ongoing study, and I don't have all the results, but I can talk you through some of our interim analysis, which is interesting. I have to say we've paid meticulous attention to the phenotyping. So I have to take my hat off to Luis in Colombia, who's been doing this, he himself has now undertaken this protocol, which is not trivial, in 750 normal subjects. So we're doing this in normal subjects, mainly students at the university. And it may not be a surprise to you that when you do this and you look at these different sensory modalities, for instance, heat pain versus mechanical pain versus pressure pain, many of them don't correlate with each other, but some do. So we're finding some interesting interrelationships between different sensory modalities. And I think, as you may predict, it's very true to say that there are some differences as well as some similarities. When we start looking at the genetics of the population, and actually in some of the analysis you can use this to your advantage, we can see that in the center there in blue, this is with a principal component analysis to try and tease out the different ancestries. You have the Colombian population, and in essence you have an input from a European, Native American, and an African lineage. We can see that mix within our population within the University of Antioquia. And I think we've kind of about halfway through this study, and on interim analysis we can see some associations when we're looking at those experimental pain levels that are reaching genome-wide level significance. And as you may predict, and a bit like I was telling you when you were just looking at correlations between those different sensory parameters, where actually these associations are quite distinct for different sensory modalities. So just as a few examples, if we look at flare area, and now this is what's called a Manhattan plot, named after looking at the Manhattan skyline, where essentially you're looking now at multiple polymorphisms across the whole genome, and each one of these columns is a chromosome, and we're then looking on the y-axis is giving you a value for significance. And one of the issues with these studies, because you're doing so many repeated testing and a Bonferroni correction, you need to get a very high level of significance before you're going to decide that it's significant at a genome-wide level, and that's that red line. And you can see right at the end when we're looking at the area of the flare, so that's a neurogenic vascular response to the mustard oil, we are seeing a significant association. And when we come to heat pain, and as I said, we're really doing this as an intermediate phenotype in terms of relation to chronic pain. When we look at heat pain, again, we're seeing a series of single nucleotide polymorphisms which are associated with heat pain thresholds. Now, I think it's fair to say, as I absolutely told you, genetics is not going to be the full explanation for pain, and each one of these is going to be having a very small contribution of a few percent to the variance that we're seeing in our population. And this is now just showing kind of at higher level of one of these genes, showing a group of single nucleotide polymorphisms which are related to heat pain threshold. And some of these genes are looking quite interesting, and some of them would have a pedigree, and some of them interact with voltage-gated sodium channels. So what we're doing now is repeating the whole study again to get more subjects, and also looking at some of the basic biology of some of the genes that we're finding using this approach. So really we've taken the idea that using a quantitative trait, hopefully by trying to understand the genetics of that, we can eventually leapfrog onto the more difficult clinical problem, or what may be risk factors for chronic pain in the general population. So I'm going to end on that point and just conclude. And I hope I've convinced you that there are rare Mendelian pain disorders, and these are related to usually gain-of-function mutations in a number of different ion channels, a number of different voltage-gated sodium channels, as well as TRPA1, which is an important transductor element. And I don't think the story will end here. I think there will be more of these Mendelian pain disorders as we get more access to sequencing and more access to these rare kindreds around the world. And I have to say, I think they're a great way of really studying the biology of the nociceptive system. They've given us huge insight into how nociceptors work, and of course they've given us huge insight as to what may be good drug targets for analgesic development, because we can really predict what may be the on-target effects of these drugs. I think it's also true to say we will see some rare mutations, probably related to the genes I've just been telling you about, that in a few % of patients will modulate risk in more common acquired conditions such as painful diabetic neuropathy, but this is just going to be in a small % of patients. And I've told you the much more difficult problem is these chronic acquired pain conditions, which are very much going to be as a result of the interaction of multiple genes having a small effect and interacting with the environment. And I've given you an example where, by using an experimental pain model, we're trying to simplify this problem as an intermediate step of moving on to chronic pain in patients. And with that, I'd like to end and just thank my lab that have done much of this work. It's been fun doing this work, and in particularly Andrea Themistokliou and Juan Ramirez, who've been really doing a lot of the clinical phenotyping on these rare Mendelian pain disorders. Greg Weir and John Dawes and Alex have been doing the functional analysis. It couldn't have happened without Luis Miguel, who I take my hat off for his perseverance in doing very detailed sensory phenotyping in huge numbers of normal subjects, Gabriel Bedoya in Colombia, and Andres Ruiz Linares at UCL, who again has been working on that Colombian study. Thank you very much.